All right. Welcome, everyone, to tonight's Wild Virginia webinar on PFAS and its effects on wildlife. My name is Jacqueline Goodrum. I'm Wild Virginia's Conservation Policy Associate, and I'm joined this evening by my colleagues, Dr. Jessica Roberts, Director of Habitat Connectivity for Wild Virginia, and Dave Sly, Wild Virginia's Conservation Director, as well as our three expert guest speakers, Dr. Anna Roebuck, Dr. Jason Hoverman, and Dr. Andrea Bonasole Alaquate. Two things before we begin. First, this webinar is being recorded. Just keep that in mind if you're uncomfortable having your name presented in the Q&A or the chat. And second, we will address questions at the end after all three panelists have presented, but please feel free to put questions in the Q&A throughout, but know that we will not be answering them immediately. And I will now turn things over to Jess to introduce our first speaker.
Thank you, Jackie. So to kick us off tonight, we're going to start with Dr. Anna Roebuck. She is an environmental chemist with the U.S. Environmental Protection Agency in the Office of Research and Development. During her academic career, she studied PFAS and plastic pollution in water and wildlife, and she earned her PhD in chemical oceanography from the University of Rhode Island. So she's going to give us a little bit of an introduction to the topic. So take it away, Dr. Roebuck.
Great, thank you so much. Let me go ahead and get my my screen share going here. Okay, and get into presenter mode. One minute. Okay, great. Could someone confirm that you see my slides in presentation mode?
I see them.
Awesome. Okay. Well, good evening, everyone. I'm so thrilled to join this virtual event. Thank you so much to Wild Virginia for putting together this great little event. My name is Dr. Anna Roebuck, and I'm a principal investigator with the U.S. EPA Office of Research and Development. I have the standard disclaimer up on the screen. I am in the Office of Research and Development, so I don't represent the rules and policies of the agency. I'm just going to talk to you about some of my research here tonight. And my research focuses on PFAS. Myself and my team, we focus on measuring PFAS in water, soil, sediment, and all different kinds of living organisms using a couple different types of techniques to better understand where PFAS are in the environment and also learn something about their impacts. So today I'll be providing you some background about PFAS as well as telling you a little bit about our work. So today we're talking about PFAS, or per and polyfluoroalkyl substances. I'll use the PFAS abbreviation through most of the presentation, but when I say PFAS or the full chemical name or forever chemicals, it's the same group of pollutants. The slide here displays an actual PFAS molecule, one specifically called perfluorooctane sulfonic acid, or PFOS, PFOS. This is one of the most well-studied compounds to date. And I know that looking at chemical structures can really invoke a lot of panic from audiences, but I think looking at the structure, it really helps hit home why these are such unique chemicals and why we have to be concerned about them in kind of a straightforward way. So bear with me for two slides here. So first things, let me point out a few things about this molecule. The most important defining characteristic of this group of chemicals is the reliance on multiple carbon and fluorine bonds, the C's and the F's in this picture that I have highlighted with a red bar. This PFOS molecule, PFOS, has 17 fluorine atoms to its 8 carbons. And so the inclusion of so many of these carbon-fluorine bonds is critical to what constitutes a PFAS, because the carbon-fluorine bond is one of the most stable bonds in all of organic chemistry and all of chemistry, really, in that it is so stable because of the way fluorine pulls on electrons away from carbon. This makes the bonds very difficult to attack and break down and contributes to why these are such stable molecules in the environment. The bond stability is, did I flip slides? Oh yeah, I did, darn. Sorry about that. Let me go back here for 1 second. I think I might have a timing on that slide. But so back to this bond discussion here. The bond is so stable and so persistent that it makes the chemicals just really difficult to break down. This is really great in terms of its application in industrial processes, which we'll talk about in a second, but also means that these molecules are incredibly persistent to the extent of they're persistent on geological timescales once they're out into the environment. And so beyond that prolific use of that particular bond, that carbon-fluorine bond, most PFAS include some type of functional group, or what we can call, say, the molecule's flare, to kind of speak generally. In the molecule that we have pictured here, we have a sulfonate group on the right-hand side of the molecule, highlighted with the red here. Many of these functional groups are acidic, or, oh man, I'm sorry, I think I have timings on some of my slides. Hopefully it stops. We will shortly find out, so bear with me a second on there. But anyways, these functional groups, they are important in that many of them are charged. So this gives PFAS the ability, or gives many PFAS the ability to associate with water. So long story short, these molecules are built to be highly stable, and yet they have some interesting chemical features that make them associate with water and travel through water systems, which is much different from many other chemicals. And all these unique chemical properties add up to the creation of many different types of these chemicals. It makes them useful for a lot of different industrial applications, and we've created so many of these. The slide here is from an older paper that tried to group all these chemicals by family. A lot of chemical kind of classes on here, but suffice it to say, we know about at least 12,000 of these chemicals. Kind of depends on how you define what constitutes a PFAS on a chemical definition level, but that's so annoying. I'm sorry, everyone. Anyways, we have a lot of these, many of them that we don't know that much about, or ones that we have recently started using or just discovered, some of which, two different families highlighted at the top in blue, those that we do know a lot about and have studied a little bit more than many of the other ones. So as mentioned, we have so many of them because they're really useful in a lot of different commercial applications. The slide here displays just a few snapshots of some of the few types of products or items that we know contain PFAS, including nonstick cookware, some outdoor gear, paints, cosmetics, firefighting foams, popcorn, food packaging. PFAS are added to all these products because they impart stability, thermal protection, oil and water repellents, lubrication, or some other attractive quality in that given product that make these really useful industrial chemicals. But the crux of the issue boils down to the fact that PFAS don't stay in all these products where they're intentionally added, right? They can enter the environment directly from production sites that create or use PFAS, migrating beyond that specific location and into the surrounding environment, or they can seep into groundwater from those production locations. They can also enter our indoor environments from leaching out of the products where we intentionally put them. They also abundantly enter the environment through human waste streams. Products containing PFAS can get discarded, and then the PFAS can leach out of those products into the wider environment through a landfill or wherever those products end up in the environment. Or since we use products that contain PFAS on a daily basis, PFAS enter our bodies and then we excrete them in our urine and feces. They then enter the water system through wastewater treatment systems or septic systems and kind of leaching out of those systems. And then PFAS can also enter the food system that way, in that we apply wastewater sludge across the country as a fertilizer that can be loaded with PFAS and then enter into our crops that way. And so the multiple pathways that contribute PFAS to the wider environment preferentially contribute PFAS to water systems, as that schematic on the last slide hopefully pointed out. As the Disney movie Finding Nemo so eloquently pointed out, all drains lead to the ocean. So when we talk about PFAS, we really think about them traveling via the hydrosphere, and they're kind of hijacking those natural water cycles in order to travel through the atmosphere, our rivers, our streams, our groundwater, our oceans. This is much different from, again, a lot of other chemicals we've previously encountered in the past, like polychlorinated biphenyls and many pesticides. They don't prefer water in the same way that PFAS do. So, and since PFAS find their way into water systems in the way that they do, we do find them prolifically in fish. The figures here come from two research efforts by colleagues of mine out of North Carolina and New Jersey, respectively, that show levels of PFAS in fish tissues by location. On the left-hand side here, we have a paper by Teresa Gillette from North Carolina State University, where they measured a bunch of different types of PFAS in striped bass from an exposed area downstream of an industrial location, as well as a control location very far away from that industrial location. And they found, obviously, that the fish closer to the industrial location had much higher levels of total PFAS and PFOS, that chemical structure I showed you at the beginning of the slide of the presentation here. On the right-hand side is a figure from a paper by Sandra Goodrow from the New Jersey Department of Environmental Protection. And this was a state effort that they published showing that different freshwater fish from different locations across the state contained very different levels of PFAS and other chemicals, ranging for PFAS anywhere from 2 to 130 parts per billion in edible tissues. We see the same variation in PFAS levels by location and species in marine environments. This is work from my team. This was led by our postdoc, Melanie Hedgespeth, which showed that variable levels of PFAS were found in marine fish, depending on their position in the food web, as well as which habitat they preferred, whether they like to hang out in the benthos and the sediment, or whether they were open water fish. And we see PFAS levels ranging from barely detectable up to about 10 to 20 parts per billion, or nanograms per gram. I highlight all these examples first because exposure identification and measuring PFAS in a given organism is kind of the critical first step towards figuring out potential effects, which is what we're talking about here today. Once we know that PFAS may be in a given species or a given family of species, the next logical step is trying to ascertain what health outcomes may be associated with that exposure. And so, as I hope those two past slides conveyed, we do frequently find PFAS in wild fish. So a large body of work has now accumulated focusing on determining PFAS impacts in fish in laboratory settings, as well as in wild populations. And this was a nice literature review that came out a couple years ago that tried to synthesize the state of the knowledge about PFAS impacts in fish. And that paper points out that we have tiers of knowledge where we have a lot of information about molecular level effects based on lab-based populations or controlled studies. And whereas population level effects are possible, we don't have as many studies showing those at this point. But we do know that those population effects could potentially impact many more habitats and locations and species. Oop, there's that timing again. But again, we know less about those. But overall, the synthesis of all this information by this group out of Taiwan found that the PFAS impact a variety of different fish species by disrupting a number of metabolic pathways. And this is mediated by sex and age of the organism at hand. At the population level, these metabolic effects mostly manifest themselves in reproductive issues, reduced fertility or fecundity, impacts in hatching or development directly out of the egg, and potentially low survival rate. And as it turns out, we readily see some of the findings highlighted in that review paper in practice in our own work. This slide shows data from another EPA scientist. Dr. Brian Clark is a colleague I work with a lot in my division within the Office of Research and Development. And Brian's team cares for hundreds or maybe even thousands of this fish called Fungulus heteroclitus, or mummychugs. I think they're also called Atlantic killifish. There's a schematic of one in the upper right-hand corner here. But this is a key forage fish found along the U.S. Atlantic coast. And we work with Brian's team to expose these fish to all sorts of different chemicals. Right now, we do a lot of PFAS work to see how they respond on an individual level as well as on a population level. Some of that most recent work has involved feeding these fish diets that have various levels of specific PFAS in controlled amounts via their fish food. And this slide shows that when we feed these fish PFAS, PFOS, and PFHXS, that's a slightly smaller PFAS that's related to PFAS. When we feed these fish those two discrete PFAS chemicals, we find that the fish abdominal fat layer gets smaller. So specifically with PFH excess, this was a more pronounced effect where all different treatment levels or exposure levels of those fish showed a decrease in abdominal fat layer with increasing PFAS level. The effect was less pronounced for PFAS, PFOS, but the highest exposure level was the significant effect here. And this is important. I threw this picture of a fat bear up there because I want you to think for a second just how important it is for wild organisms to have reliable fat stores. Fat stores are critical for timing migration and hibernation and reproduction. And so a chemical impacting fat reserves and overall metabolic pathways is a real eyebrow raiser. It's something we really need to pay attention to. I want to stick a pin in that and actually move on and change directions completely and move to team bird. I want to talk about the types of studies that I work on a little bit more rigorously. And so I'm a chemist. I'm not a toxicologist at all, actually. So I don't perform those controlled lab studies. I do Brian's measurements. I don't deal with all the fish. But I have found a rather effective way to link PFAS exposure to impacts by conducting observational studies. And just so happens I really like birds. I'm a big bird watcher. So much of my research revolves around wild birds. And in these studies, I specifically work a lot on seabirds, the species pictured here. It's the great shearwater. We measure as many individuals as we can, and we collect as much information as possible about each individual. We measure their wings, their beak size, how big all their organs are, just everything possible that we can during a dissection or a necropsy procedure. And then we relate that information back to the levels of PFAS that we measure across different tissues in these organisms. And we try to just use everything, beak to claws. We take measurements on it in terms of morphometric information as well as PFAS information. And so this allows us to understand potential associations between PFAS levels and health outcomes in wild animals exposed to environmentally relevant levels of PFAS, which sometimes can be lacking in lab studies, right? Lab organisms are subject to very simple exposures in most cases, meaning that they're not necessarily exposed to the mixture of PFAS that is found in a realistic environment. So these observational studies try to speak to that a little bit with, of course, a lot of confounding factors behind it, but we'll talk about that a little bit later. But regardless, this is the type of study that is really my bread and butter. And in those types of studies, we find something that is really similar to what Brian sees in his controlled lab studies in fish, right? By measuring hundreds of these seabirds and other types of seabirds as well, we find that PFAS, PFOS, those concentrations in the liver of these birds were inversely related to phospholipid levels in the liver of these birds. And phospholipids are just a type of fat. It's really important overall for cellular structure and overall metabolism. It's a family of fats, we can say. But these results suggest that PFAS could be associated with decreased phospholipid levels in wild birds. These results, again, have big implications for wild populations. The species that I particularly work on, they live off the coast of Antarctica in this really remote location, and then they migrate all the way up to the elbow of Massachusetts during the summer. And so they really need finely tuned fat reserves in order to make that happen. And so we are seeing these associations in lab studies as well as in our wild populations via observational studies. In addition to that, it's not just fat layers or specific fat concentrations that may be associated with PFAS levels. We also found that great shearwaters were generally smaller as approximated by beak size and wing length. They had smaller fat layers, as I mentioned, and their organs were smaller. They had smaller livers and smaller kidneys compared to birds with less PFAS. On the x-axis here, this is just another PFAS, a specific type of PFAS. This is not a molecule I've showed yet, so I just wanted to call that out. Whereas on the y-axis here, that is the mass of the bird's liver. On a molecular level, these associations could relate to the capacity of PFAS to modulate hormones as well as modulate metabolism. So a lot to be discovered about the exact mechanism of what drives all this. But again, we are seeing these associations between PFAS exposure and health outcomes in wild populations. So with that, I'm getting, I think, closer to my time to call it here. And I wanted to call out a few things in summary. So we do find that research shows associations between PFAS and various health outcomes, both in laboratory controlled study and in fish. It's tough to put a finger on exactly what's happening in relation to PFAS and wild animal populations, because wild animals are exposed to so many different stressors, chemical, climate, food availability. There's so many different things stressing out wild animals. So observational studies are getting us some sense of ideas about what is impacting those wild populations, but still a lot more to be done. Some of that work is additionally challenged by the fact that we are not specifically monitoring one given species for PFAS. A lot of the monitoring of PFAS is still being established. We don't know which species to monitor. We don't know where or when to do that, because PFAS levels can be impacted by reproduction. So there's a lot of variability between a lot of the measurements we're using to figure out impacts, right? This is further exacerbated by the fact it's kind of hard to measure PFAS. It's not an easy task. There's not a lot of validated methods available yet. EPA just came out with a validated method for 40 of those compounds, but there's so many of these different compounds in the environment. We don't know how to measure them all that well in biological tissues. Yet monitoring is key. As I mentioned at the beginning of all this, in order to be able to start talking about impacts, we need to be figuring out where PFAS are in the environment. The agency is making a lot of commitments to continue tackling these questions. I want to point out that a couple huge things happened this month in terms of EPA progress on PFAS. The agency released drinking water levels, as well as listed twos of the PFAS substances under the Superfund law, which is a big deal. I would encourage you to Google those terms, use this QR code to check out those new regulations, or draft regulations, I should say, and check out the EPA's strategic roadmap. A lot has been happening in the agency related to these compounds. So with that, I think I'm just at my time. I would love to hear comments and questions at the end. And my contact information is here if you have any specific questions beyond what we can cover today. But overall, thank you for having me, and thanks for your attention on PFAS.
Thank you. That was wonderful. And we will provide some supplementary material in an email after the webinar. So don't worry about copying down her email just now if you want it. So moving on, we're going to go to Dr. Jason Hoverman. He is a professor in the Department of Forestry and Natural Resources at Purdue University. Since 2016, he and his lab have worked to understand the effects of PFAS on freshwater aquatic communities. Jason received his PhD from the University of Pittsburgh. So take it away, Dr. Hoverman.
Great. Get this going here. You see it all right? Great. Well, thank you so much for the introduction, and thank you to Virginia for inviting me to do this. It's always exciting to kind of reach out to new people about some of the research that we're doing. So what I'm going to do today is talk a little bit about some of our work looking at the effects of PFAS on wetland communities, and especially on amphibian populations. So as you all know, wetland habitats are very diverse ecosystems, and as was kind of just covered, these wetland habitats also receive runoff from terrestrial landscapes that oftentimes are having a variety of different types of contaminants. But again, more recently, we've come to realize that PFAS has been a big concern when it comes to wetland habitats. So what I'm going to do today is just talk a little bit about some of our work we've been doing since about 2016. Give you a little bit of a few snippets of some of the work that we've done. So I'll start off with a field study. Again, to provide some environmental context, this has been done at a heavily contaminated site in Michigan. And then we use that information to basically feed into some of our laboratory experiments where we assess things like acute toxicity, so how much mortality is caused, following exposures, looking at chronic toxicity on things like growth, development, and reproduction. And also, I'll talk a little bit about some of our work looking at how PFAS can actually lead to an increase in disease risk, and especially infectious disease risk for amphibian populations. So jumping right into our field study. So Clark's Marsh is in Michigan. It's right next to Worth-Smith Air Force Base, as you see here. So for decades, this Air Force Base was using these firefighting foams containing PFAS in training exercises. And as you can imagine, runoff ends up going off into these wetland habitats. So what's happened is there's a PFAS gradient going from really high right next to the Air Force Base going to lower as you move from this A to C in this schematic. So we were really interested in trying to understand how PFAS is distributed not only in the water and the sediment, but basically throughout the food web at this marsh. So we basically went to this marsh, collected a variety of organisms, all the way up to our top predators in the system, which include things like fish, turtles, as well as snakes in the habitat. So just really jumping right off into some of the data that we're finding. So what you're seeing over here, this is the PFAS distribution across our different kind of compartments or taxa. So we start off with things like water and sediment here. We also sampled some terrestrial plant species as well, kind of surrounding that marsh area. And then we have a variety of aquatic species as well. So a few things to kind of point out here. First off, we're actually finding 14 different PFAS at this marsh. So again, when we're thinking about PFAS, it was kind of previously mentioned, right? There's over 10,000 different types of PFAS. This is especially the case when it comes to these firefighting foams, where they use different kind of components and mixtures in those firefighting foams. So you end up with a broad diversity of different PFAS in your samples. But when you look at kind of the breakdown of these PFAS, again, we see PFOS in particular is one of the more common, so like this dark green coloration, especially for the aquatic taxa that we have present in this marsh. We also see that PFHXS, the lighter green colorations that you see here, is also relatively common as well, especially in things like crayfish in our marsh. But what was really interesting as well is the terrestrial plants. They have a very different PFAS profile. So in particular, you're seeing this yellow coloration here. That's PFHXA in this case, which we might expect because plants are going to have very different uptake rates of PFAS. And again, depending on how that PFAS basically sticks to the soil and the sediment, that can influence how it's taken up by other species within that wetland as well. So this gives you an idea of just how many different PFAS can be at some of these sites. Let's jump into the concentrations we're actually seeing the taxa in this marsh. So what you're seeing here are the invertebrates in gray, the amphibians in green, and the fish in red. And what we have here, this is the gradient going from very close to the Air Force Base, CM1, to far away from that Air Force Base in CM3. And what you're noticing here is some variation, right? So first off, as you move away from the Air Force Base, we start to see a decrease in that PFAS concentration, right? So as taxa are kind of picking up those PFAS, that's taking away, and also you're getting dilution as you move further away from the Air Force Base as well. But the other thing to kind of highlight here is you see really big differences between the fish in the community and then what we see in the invertebrates and the amphibians. And what that is suggesting to us, right, is that we're thinking about the accumulation of these compounds, that these higher trophic levels are picking up higher levels than we're seeing in the invertebrates and the amphibians in that food web. So I'll come back to that in the next slide as well. But importantly, what this data allowed us to do is get an idea of what these guys are being exposed to. So we have a range anywhere from 10 to about 1,000 parts per billion across our sampling regime. So a pretty broad variation, even within just this one marsh. It's very close to the Air Force Base. But again, we had this question about, okay, let's look at this accumulation through the food web. So what you're seeing here are various PFAS that we found. This is the trophic position. So we use stable isotope analyses to figure out where a taxa kind of fits within that food web, going from, again, your plants, which are going to be very low, and then your fish and your reptiles being around the 4 in this case on the graph. And then what we're graphing here is the PFAS concentration in the different taxa, with the different taxa being different colors. So kind of walk you through what we're seeing here. First off, if we're looking at kind of these top 2 rows, for example, looking at PFOA, the correlation between the trophic position and the PFAS concentration is actually pretty weak. We don't have a very strong relationship there, right? It looks kind of like a shotgun blast. But when we look at things like PFOS in particular, right, we have a much stronger correlation, suggesting that this PFOS, right, is more bioaccumulative. So as you get higher in the trophic level, these species are picking up more and more of the PFAS. And again, if you're looking at total PFAS, that's basically paralleling what we see with PFOS, right? Because it's the most dominant that we have in terms of the food web. And we also calculate what's called a trophic magnification factor, and that came out to about 6.7. So anything that's above 1 shows significant magnification. So in this case, we're seeing very strong magnification. And sorry, my printer just decided to go off back behind me. But really the take home from this data is that, again, diet is a really important source of exposure, right? So as these animals, especially fish, reptiles, are eating these different taxa, they're picking up the PFAS burden from those animals and they're taking it up into their bodies, right? So that's one of the main concerns when we have, or that we have with PFAS. Okay, so the next thing we want to do is use this information to start to explore the ecotoxicology associated with PFAS. So really the next step for us to determine whether or not these concentrations that we see, especially these contaminated sites, might be influencing mortality, right? So if we see these levels in the environment, are they going to be directly toxic to our focal taxa? In our case, that's amphibians. So what we've done is a variety of what are called lethal concentration 50 experiments. And what these are is basically use a broad range of concentrations. So you can just kind of see this in this little schematic here. So this broad range of concentrations, and your response in this case is mortality. And what we're trying to figure out is at what concentration do you see 50% mortality? So basically what you do is just kind of draw a line down here, and that's your LC50 value. So the lower those values are, the more toxic a compound is or a chemical is. So what we've done to date is tested 9 different amphibian species in the larval stage. So that included salamanders, toads, and frogs. And we focused on 2 PFAS, so PFOS and PFOA, which again are kind of the most commonly talked about when it comes to PFAS. And what we found is this. So on the x-axis here, we're seeing our different species. So we're starting off with salamanders, go to toads, and then a few other anurans in the group here. We have PFOS at the bottom, PFOA at the top. And again, what we're showing here is the LC50 estimate for those different species. Okay, so what you'll notice right off the bat is that PFOS is about 8 times more toxic than PFOA, right? So we see lower values in this panel than what we see for this panel, okay? Secondly, we see a lot of species level variation, right? So if we're looking at our salamanders, for example, they were more sensitive to PFOS than our anurans, so our frogs and our toads in our sample. But what I want you to take home from this, though, is the actual concentrations here. The toxicity values that you see in this figure are way higher than what we'd find at our contaminated marsh, for example, in Clark's Marsh, Michigan, in this case. So in this case, these milligrams per liter is essentially parts per million. But what we're seeing actually out in the field is parts per billion. Okay, so what this tells us is acute toxicity is not likely in the field. So if these amphibians come down to breed in these habitats, they lay their eggs, or tadpoles develop, those concentrations aren't going to directly kill our amphibian populations. Okay, well, that's good. But the bigger question we want to know is what are the potential adverse effects when you have exposure levels that you actually see in the field, which again are going to be much lower than it would take to be directly toxic. And as is previously covered, we know that PFAS, for example, are known endocrine disruptors. So they influence things like growth, development, reproduction, and a variety of different taxa. So we need to kind of explore this, or more long-term experiments at these lower concentrations that mimic what we're seeing out in the field to figure out if we're having any effects on these types of response variables for amphibians. So over the past few years, we've done a broad diversity of experiments. We've used a diversity of species, northern leopard frogs, American toads, tiger salamanders. We've explored different PFAS. So you can see some of those listed here. We've looked at different life stages of these amphibians, so the larval stage or the tadpole. We've let them metamorphose and then tested the subadults or metamorphs. And we've also looked at different exposure routes. So just simple aquatic exposures through the water. We've exposed them through sediments or substrates. And we've also done oral exposures where we actually have dosed their food and then fed that to the amphibians to look at how that might impact them as well. Okay, so I'm not going to cover all those studies. It's a pretty large number of studies. I'm going to focus on 2 of those today to give you an idea of some of the effects that we're actually seeing. So the first of those is going to be an aquatic test. And these aquatic tests are really simple. Basically, you set up little tubs, you put your amphibians in those tubs, and then you can add PFAS at different levels to those tubs. So in our particular case, we were using 10, 100, and 1,000 parts per billion or milligrams per liter. So again, these are concentrations that you would find at a contaminated site with AFFF exposure associated PFAS. Okay, and we let these experiments run for about a month. We take them down and we look at things like, how fast they grew, how fast they developed. What I'm going to focus on today, though, is this idea of body condition. So this, again, parallels nicely with what Anna talked about as well, in terms of body condition being a really important factor when it comes to long-term fitness of amphibian populations. Okay, so you can basically think about it as our body condition index when you go to the doctor, right? They want to know what your BMI is. Similar thing for amphibians. Okay, so walking you through some of this data. On the x-axis here, you're seeing the PFAS concentration. The different colors are the different PFAS that we tested. So orange is PFOA, red is PFOS, the green is PFHXS, and the blue is 6,2-FTS. We have northern leopard frogs on the left, in the middle are American toads, and on the right are salamanders. And the data here are means plus or minus a confidence interval, so that's why those error bars look really big on this figure. But the first take home from this is that actually for American toads, they don't seem to care too much. We see no significant effects when it comes to these toads. But when we look at the other two species, what we see here is a significant reduction in the body condition. This SMI is decreasing. So I'll just focus on leopard frogs here. So if you look at all these colored dots, they're pretty much resting below our control, which is the open circle right here. Okay, but there's a few more bits of data to kind of pull from this as well. The second is that we see significant variation across how the PFAS impact this amphibian, right? So we see for two of them, PFOA and PFOS, we see moderate effects, right? But for the next two, we see that the effect is basically twice as great. So we see basically a doubling in terms of how far that scale mass index reduced relative to those other two PFAS, right? So we have some variation across there. And the other thing I want to highlight here is that our strongest effects aren't necessarily associated with those PFAS that are the most bioaccumulative, right? So we hear a lot about PFOS, right? We've heard that in the previous talk. You saw this in the Clark's Marsh data where we have lots of PFOS and that's accumulating through the food web. But also for things like 6,2-FTS, which doesn't tend to accumulate as readily, we also see significant effects for this PFAS as well. So that's something to kind of note when it comes to PFAS. Okay, so the other bit of data I'll talk about are these terrestrial exposures. So in this case, what we did is devise an experiment where we could actually dose the food of our salamanders, which are the crickets, with PFAS, and then feed those crickets to the salamanders to do our exposures that way. So what we did here is basically you can basically rehydrate potato flakes with different concentrations of PFAS and then use that as the food source for the crickets. And then we also had a little sponge here, which is the water for the crickets, which is dosed at these different PFAS levels as well. And basically what you do is after you have dosed the crickets, you feed those to the salamanders, run the experiment for about 30 days, and now you can look at dietary exposure to PFAS for these terrestrial amphibians. So the first thing I want to show you with this experiment is actually the body burden data. So how much was accumulated in the crickets as well as the salamanders. So if we look at the crickets, as we'd expect, right? As you increase the dosage in their food, the crickets end up picking up those different levels of PFAS, right? So we have this nice dose-response relationship when it comes to the crickets. But when we fed those to the salamanders, we see pretty significant variation in how much they were picking up of these different PFAS. So for example, 6,2-FTS didn't really transfer to the salamanders, right? But we have two that are kind of intermediate, so that's PFOA and PFHXS, and then high accumulation, again, not unexpectedly with PFOS, right? So basically matches the patterns we typically know for most of the PFAS and how they accumulate within taxa. So next question is, okay, what impact did that have on our scale mass index, our body condition of their salamanders? So that's what you're seeing here. So basically same setup as I showed you for the larval exposures. But again, kind of breaking this down a little bit. For PFOS in particular, we have pretty strong effects, right? So these red squares are all resting below our control treatment. When we looked at PFOA and PFHXS, what we have are what are called non-monotonic responses. So all that means is that's basically a nonlinear response. So we see significant effects at our lower concentrations. So you can see, again, our green and our orange or yellow in this case are below our control. But when we looked at our highest concentration, there was no effect. And we actually see this pretty commonly with a lot of our PFAS experiments where we have effects at our lower concentrations. Those kind of disappear at our higher concentrations, which is kind of interesting. And then we turn to 6,2-FTS. And in this case, we find no effect, right? So actually lots of variation here compared to what we're seeing at the larval stage. And again, comparing across different species as well. Okay, so the last thing I want to talk about is some of our work looking at the kind of interaction between PFAS exposure and disease risk. And if we think about PFAS, right, it's occurring in a natural habitat where there's lots of different factors out there, right? Where there's competitors, there's predators, and importantly, there's parasites. And we know that many contaminants can have an immunosuppressive effect on species, right? We know this for pesticides in particular. The expose an animal to pesticides that suppresses immune function and increases the risk of infection. And unfortunately for PFAS, there's not a lot of information out there on that. So for the past few years, we've been trying to kind of fill that void, especially when it comes to amphibian populations. So the model system for this, I'll just kind of briefly talk about this, involves trematodes. Trematodes are a parasitic worm that gets into amphibian larvae, and they have this complex life cycle. So they use birds as the definitive host. That bird will defecate into a wetland. There's a stage that seeks out a freshwater snail. And then that snail basically produces all these cercaria, which is another free-living stage of the parasite that seeks out the amphibian. And our particular parasite, in this case, is echinostomes, and they settle in the kidney tissue of the amphibian. So all these little circles that you see in this tissue, this is a kidney tissue, are all parasites of these amphibians. So just really quickly, we did this two-phase experimental design where we had American bullfrogs exposed to different PFAS treatments. You can see those listed here. Just very briefly, we did different PFOS exposures. And we also had some mixtures of PFOS as well to try to match the mixture that we actually saw at Clark's Marsh, Michigan. So I won't focus too much on that. But what we did then is after they were exposed to PFAS, we took them out and exposed them individually to these little parasites to see how many encysted within the tadpole. And what we found was that indeed parasite loads were much higher in our PFAS exposures than they were in our control. So again, here's our control, and all of our PFAS treatments had higher parasite loads than what we saw in the control treatment. So it ranged anywhere from 42 to 100% higher in those treatments. So what this does suggest is that immunosuppressive effect of PFAS can have an effect on infectious disease risk for amphibian populations. So just kind of bring this all home for you. Some of the summary of what we're seeing, we see significant trophic magnification in these wetland systems, and especially those higher trophic levels are picking up PFAS, and that's probably a lot really driven by that dietary exposure for those organisms. But for amphibians in particular, we don't expect acute toxicity in these habitats, but what we do expect are these potential chronic effects on things like growth, development, and body condition. And we also see significant variation depending on what amphibian species we're looking at and also what PFAS we're looking at as well. So that's really an open area of investigation. And then lastly, I think really critically as well, another potential chronic effect is this chronic effect on immune function and the cascading effects there could be on disease risk. And that's especially true for the amphibian work that we've done so far. So the last thing I'll leave you with are some of the unknowns. And when it comes to PFAS, there are still a lot of things that we don't know of. And Anna kind of spoke to the population level responses. At this point for amphibians, we don't have the long-term data to know whether or not these exposures are leading to population level effects. So that's definitely something that we need to explore. And along those same lines, we don't know a lot about the effects on communities and ecosystems. Right? These are very complex, diverse systems. And that's really an area that research needs to kind of focus on and looking at these indirect effects and trophic cascades initiated by PFAS. And the last thing that I'm really interested in is looking at potential evolutionary responses. If we think about PFAS, it's been used for decades. That's a lot of time. And especially from an evolutionary perspective, some of these taxa might be evolving evolutionary responses to these chemicals as well. So I think that's another interesting area of exploration. So with that, just going to thank collaborators. A large number of undergraduates, graduate students, postdocs, as well as PIs have helped out on this project. And we've been fortunate enough to receive funding from a broad diversity of resources, including the Michigan DNR, USGS, EPA, the CERTUP program, and also Purdue University as well. So with that, thank you, and happy to address questions at the end.
Thank you, Dr. Hoverman. So last but definitely not least, we have Dr. Andrea Bonasole-Alquati. He's a professor at the California State Polytechnic University in Pomona. His lab is researching the concentrations of PFAS in bird tissues, both directly and through meta-analysis studies. And they're also conducting studies on the biomagnification of PFAS in marine ecosystems. And he received his PhD from the University of Milan in Italy. So take it away, Dr. Bonasole-Alquati.
Thank you very much for the introduction. Hopefully you see the presentation in presenter mode. Perfect. Hi, everyone. As Jessica was saying, I'm a professor at Cal Poly Pomona, and it started to follow the great presentations that my colleagues did. And so to switch approach a little bit, I'll both, in a sense, zoom in into a single compound and also provide an overview of the studies that have been conducted so far, focusing specifically on birds, just because birds sort of continue this tradition of being used as excellent sentinels of environmental problems, and PFAS are just one of the emerging ones. Although, again, as I will show, and they've been studied in wildlife for a few decades. So the approach that I'm taking with this is related to, is called the systematic review and meta-analysis. And again, it consists in the reuse of the existing scientific literature, the existing results, existing investigation in a more combined, so systematic context. So in order to provide not only an overview, but also new insight into the problem. And so I thought this was a good occasion for us to review what is known, at least in terms of exposure, because as my colleagues pointed out, we know a lot more so far in terms of exposure of wildlife than we know about the effects. So we have very good reasons to believe that, again, these effects exist, and especially because of lab tests, but increasingly, as Dr. Hoverman and Dr. Roebuck demonstrated, also because of experimental tests and tests that are conducted under realistic ecological conditions. I wanted to start by acknowledge that much of the heavy lifting on the work was done by an undergraduate, brilliant undergraduate student in my lab on this specific aspect. In my lab, I also complement sort of these systematic review approaches with a sort of on-the-ground study of PFAS concentration and exposure of birds and effects associated with exposure. But yeah, Raul Flamenco, I need to acknowledge him because he did much of the work and then moved on to greener pastures. Now at the National Academy of Sciences after graduate studies at UConn. And so I'll limit the introduction. We talked about the fact that, of course, in focusing on PFAS, I don't mean to diminish the really broad dimensionality of PFAS as an issue. There are an estimated more than 10,000 compounds and coming from a variety of industrial and consumer and military uses for all these substances, and their exposure, so their dynamics in an ecosystem as well as their effects are to some degree expected to vary. Although again, there's a huge gap in our understanding of these dynamics. Though again, Dr. Hoverman provided very interesting data about some of this variation. Again, from many uses of these compounds in the environment, we have the expectations and even the data to estimate that wildlife, again, focusing on birds here, have been exposed. And we've known this in a sense for decades because the first investigation started really in the 70s and 80s. The first quantifications of exposure to wildlife. And just to give you a sense of how much these estimates may vary, from a single paper published in 2001 by Cannon et al., closely related species demonstrated an incredibly high concentrations of PFAS in the case of Brandt's cormorant in San Diego, and sort of much lower and yet fairly high concentrations in double-crested cormorants from the Great Lake region. So again, there's extensive variation here among the studies. And then, of course, this is just a set of two closely related species, but if we start to expand it, we find additional variation. And of course, the catch here is that not only we're dealing with different species, but we may also be dealing with estimates that differ for how they were collected and when they were collected. And so if one looks at the literature and really asks the broad question of how much birds are exposed to PFAS in general, and then narrowing it down to PFAS just because it's the one that is a legacy contaminant, the best studied one, we expect, of course, that there's interspecific variation, so variation between species that's due to the fundamental natural history differences of the different species, but also depending on how and when they were studied. And so here you have an example of those those estimates that I provided you. They may come from different tissues, blood or eggs, or they may be coming from different time in history. And only a few studies, honestly, have the ability, logistic ability and the funding ability and the so-called manpower to conduct a study to span decades to understand those temporal dynamics and that sort of answer our question, address our worry that these persistent pollutants may be accumulating in the environment and increasing in concentration in the environment. So a few such studies do exist. And they did find, as is the case of this study from Sweden in the peregrine falcon, you see two different PFAS here, perfluorohexane sulfonate and PFAS that are, as you can see, sort of have increased through time. So in the decades from the 70s through the early 2000s, have increased exponentially in the blood of peregrine falcons. And of course, peregrine falcons are these top predators in their ecosystems. Vice versa, other studies have found that around that same or spanning part of that same amount of time in other species, such as this herring gull, the concentrations of perfluorinated carboxylate and perfluorinated sulfonates have decreased, and sort of that possibly thanks to the phasing out of some of these substances. PFAS together with PFOA were voluntarily phased out of production in the early 2000s. So again, there's variation that we need to make sense if we want to address questions about how much birds are exposed and what ecological, ecotoxicological risks they experience. And again, that's in the context of birds serving as sentinels of the environment. Not I am, like Dr. Roberts was saying, I'm totally a bird nerd, and I'm very passionate about birds, but I also promote the argument that they're really sentinels, and the effects that they experience, which is at the individual level as well as ultimately potentially at the population level, they're not expected to be substantially different from the ones that us as vertebrates would also experience. And I think this was also mentioned, some of the effects that are in terms of metabolic effects, as well as immunological effects, as well as endocrine disrupting effects, they're expected across all of these classes of vertebrates. So when we task ourselves with understanding all of this variation, we can of course expect that dietary differences or diet differences would predict variation, just because we know that chemically these pollutants tend to have a biomagnifying and bioaccumulative behavior, and this is also something that again was illustrated before. But we can also expect, again, that some of our own conclusion may depend on the experimental approach that we took and the particular tissues that we considered, as well as they may depend on some physiological aspects of the birds. Finally, they may depend on the geographic location. In particular, I don't mean by that any local source, but rather by the dynamic of generation and input and of these substances into the environment, as well as the presence of potential sinks of these substances in our environment, so on our planet. So just to make some of the hypotheses as we sort of compile this evidence, our hypotheses are therefore that PFAS will be found at higher concentrations in birds that occupy higher trophic levels as a result of this biomagnification, that it will be higher in longer-lived species, and potentially also in species that tend to reside at those more highly contaminated areas of the globe throughout the entire year, as opposed to escaping to, for example, through their migration to lesser contaminated area. We may differentiate studies that have used blood from those that have used eggs or liver, because these different tissues respond differently to exposure. There's a short residency of pollutants in circulation, but pollutants like PFAS tend to accumulate in liver. Vice versa, they may also be offloaded to the eggs, which leads also to the prediction that the two sexes will differ in how they're exposed to PFAS. But we may also predict that they will be higher at sources. With sources, I mean the latitudes where they have historically been produced and used the most, which means like the industrial north, essentially, of our planet. Whereas, but we know that they tend to concentrate at polar regions because of transport, and that works as a sink. So we may also expect that there's a trend for an increase at those latitudes. So again, I won't talk about the methods for compiling evidence from all these studies, but essentially it's a lot of reading of a lot of papers. So we started with a search of several hundred papers that we screened for fitting of our criteria for inclusion that sort of had to sort of demonstrate quality and demonstrate sort of that they were interested in quantifying the PFAS, not just assessing, for example, presence or absence or detection of the compound. But throughout this process of review, we ended up with a few hundred, about 500 estimates of concentrations of PFAS in about more than 100 different species of birds from more than 70 studies, representing sort of a great diversity of animals and a great diversity of associated niches and environment. So we started by mapping this onto the globe. And so the first thing that you see, if you look at the distribution of these estimates, is that the vast majority of them fall into the temperate region of the world. So they're quite distributed throughout. Here on this map, the width of the circle, the radius is proportional to the documented concentration of PFAS. And by the way, I should say, this is an interactive map that you may feel free to browse yourself right now. If you're familiar with QR codes, feel free to do that. And ultimately, the data set will also become public and usable in this same way. But the interesting part of this is that it points out that there's a gap in our knowledge about our global knowledge, because the majority of our estimates come from the places where you would expect, in the sense that these are the places in the temperate north and industrial and Western civilization that where most of the research is conducted. Even so, we may ask what the trends is documented, what kind of trends are documented for the various, separated by the various tissues for the the PFAS concentrations that we extracted from these studies. And what we see is that blood concentrations, this is perhaps one of the few good news we're going to hear today, that PFAS concentration have declined in birds throughout the last couple of decades. But again, tempering this, the optimism, they have instead increased. So here you have on the x-axis, I should say, you have the ear, and on the y-axis you have the PFAS concentration. And there's extensive variation in these concentrations. So to account for all of this extensive variation, what you see is an axis that is so-called logarithmic, on a logarithmic scale, meaning that every change in from one tick to the next is a tenfold change in concentration here. So again, extensive variation that exists, and a linear trend corresponding to an exponential increase through time, or decrease. But when it comes to liver, again, concentration have been increasing. For eggs, instead, the concentration has been fairly stable, although I should point out that in general, concentrations in liver and eggs turn out to be much higher. So here I'm just as a reference putting the maximum concentration detected in blood by those studies, and you can see that a good number of estimates in eggs and liver exceed estimates in blood. That when it comes to variation among species, we can see that, again, we had a clear expectation based on the niche and the diet that different species have, under the assumption, under the hypothesis that, again, these are biomagnifying compounds, PFAS are, we would expect birds that occupy higher trophic levels to have higher concentration of PFAS. And this is indeed what we find where piscivorous and carnivorous birds, here exemplified by a mirror and peregrine falcon, as well as opportunist birds and birds that are sort of synanthropic, that live in cities like gulls and feed on our sort of waste products, tend to have much higher concentrations than, say, herbivorous birds or birds that have a diet based on insect or a mixed diet. Now, when it comes to where the concentrations are highest, one of the places, again, our expectation was that in the temperate north, where production has been historically located, the concentration would be higher. And in fact, that's essentially what we see with temperate, again, extensive variation, but temperate locations are associated with higher concentrations on average than polar location and tropical locations. However, this sort of depended on the tissue that we consider. We're not going to enter too much of the statistical details here, but the idea is that whatever difference we detected sort of depended on what tissues we consider, and this difference was especially true for liver, but not so much for blood. One thing that, however, distinguishes the two locations is that while the northern temperate region concentrations have been fairly stable, they have been increasing extensively and sort of fast in Arctic region. In fact, not only they've been increasing exponentially, especially when it comes to liver concentrations, what you see in the right panel here, but the rate of increase has been speeding up, meaning that, again, probably due to transport and the Arctic region functioning as a sink, concentrations and exposure to Arctic breeding birds is increasing really fast. The very last thing I wanted to mention is the idea that, again, in addition to what aspect of the diet or what aspect of the food web or link in the food web birds occupy, there are many other ecological characteristics and life history characteristics. I'm just going to present one here because there's extensive variation among bird species in their longevity. And we're talking about substances that are bioaccumulative, meaning that they would sort of be sequestered throughout tissues throughout the lifetime of an organism. And so we expect that for those tissues that have and bioaccumulative dynamic, there will be bioaccumulation. This is specifically an expectation for liver. And in fact, we found that in the liver of birds, we find higher concentrations of PFAS the longer the lifespan of a particular species, indicating again that some of the variation that we may detect and some of the considerations about who's most at risk from PFAS exposure may be in the details of the life history of these animals. Just to summarize really quick, there's again extensive interspecific and ecological variation in how birds are exposed to PFAS. Again, focusing just on a single one, just because it's the better study one that gives us argument for interpreting these variation in exposure and in ecotoxicological risk. And extensive variation exists also for the dynamics between tissues. And so we should interpret things depending on how estimates were obtained. Birds are good sentinels, and a very good examples of this is the fact that Arctic species tend to be this, once again, as for climate change, they tend to be this very sensitive, very exposed to anthropogenic pressure, even for PFAS. And in general, ecological knowledge can really contribute to environmental toxicology, and I'm happy to explore this further with you. Thank you so much for your attention. I went a bit long, but there will be so much to say. I look forward to your questions. Thank you to the organizers for organizing this. And again, one big thanks to Raul Flamenco for help with this study.
Thank you so much, Andrea, and thank you to all our speakers. We are over time, so if you have to leave, we will be following up with you with the recording of the webinar and other materials. But we do have a couple questions. I and others started answering them in the Q&A in the interest of time. But we do have a couple that we will go ahead and address before we end. The first one is for Dr. Hoverman. Has Purdue informed the Air Force Base of your data, and have they changed their behaviors?
Yeah, so kind of the background on that is, so we got support through the Michigan DNR for that project, and they've been in close contact with the Air Force about all the activities there. So there's been tons of sampling at that marsh. So we provided a report to the Michigan DNR about the levels, distribution, those types of things. They're no longer using the PFAS there, but they're still trying to figure out how to clean up some of the issues associated with the contamination that was initiated at the marsh. Basically, or because of the Air Force Base around the local community. So there's been things like warnings for fish consumption in particular. So there's signs to warn the general public about the problem and not to consume fish. At this particular marsh as well, a couple of the white-tailed deer actually came back positive as well for PFAS levels. So very similar to I think Maine also had some issues with that as well at some of their contaminated sites. So everyone's aware of the situation for sure.
Great. Thank you for answering that question. We have another question about the bioaccumulation effect. Does a vegan diet, lower trophic level, minimize the total PFAS concentration? I assume that means in a human vegan diet. Who wants to respond?
In principle, yes. And to what we know from epidemiological studies is that the largest exposure, the source to humans is through the diet, as opposed to inhalation and direct thermal exposure. And so in principle, what we eat definitely contributes and because of the expectation that these compounds would biomagnify, of course, eating at a higher trophic level, such as eating tuna or eating meat of, especially again, high up the trophic web, may be driving exposure up.
Thanks for that response.
Any specific study.
Yeah,
So
okay.
totally interrupting and jumping. But yeah, I don't know of any specific study, but the expectation is definitely that way. Yeah.
Great. And our last question is, has any research been done on viable solutions to filter PFAS from water that flows into the watershed or wetlands specifically? This person proposes mycelial mats as the solution. Has there been any research done on that or other?
Yeah, I could field that one. My Zoom is telling me my Wi-Fi is breaking up a little bit. If I'm breaking up, I apologize. I'll type a response as well. PFAS destruction technologies have blossomed over the past 3 to 5 years. There's a lot of work being done to explore oxidation technologies, sonolysis, incineration technologies, UV degradation. I'm not specifically familiar with work being associated with the fungal mat technology, but a lot of these approaches are being applied at a drinking water treatment level and a wastewater treatment level, rather than a watershed level. That question of scale is not yet being tackled. We're focusing on getting it out of our point sources first. But maybe there's work there. I might just not be familiar.
And that segues excellently into Dave Sly is just going to speak very quickly on how you can learn more about what Wild Virginia is doing to advocate for stopping PFAS pollution at the source. Dave, could you speak to that?
Sure, I can be very quick. First of all, since you attended this, you're going to get follow-up information like Jackie said. Just please follow us. Check our website, wildvirginia.org. What we really are focusing on is stopping, where we can, stopping PFAS from entering the environment. Specifically, there are regulatory systems to control discharges of wastewater, discharges from industries, application of PFAS through land application of sewage sludge, things like that. Those are things we can get our hands on right now, and so that's why we start there. That won't be the total solution by any means, but it's an important one, and we need your support to help with that. So please follow us.
And thank you everyone for being here. Thank you to our excellent panelists, Dr. Roebuck, Dr. Hoverman, and Dr. Bonasoli-Alquati. And we are way over time, so we are going to end now, but thank you all again and have a good night.
Thank you.